Okay, now from Houston Methodist Medical Research Institute, Santosh Helekar is going to tell us about a fascinating device that he's invented that seems to be measuring proximity to consciousness.
Thank you. And I want to see how this works.
Up here. Take this.
Well, yeah, sure. Good afternoon. I would like to thank Stuart and Thomas and Abby for inviting me for this, to give a presentation on my wild project, which is very surprising, and it all happened within the last 18 months. So I haven't published anything, but this is sort of preliminary data in a sense, but it's compelling enough to us at least, so that we want to share it and get feedback on it. So what I'm a non-revolutionary, I have been a non-revolutionary neuroscientist working on Stuart's cartoon neurons for a long time, but this happened, and so I'm kind of now convinced that Stuart is right, and Sir Roger. So what I'm going to talk about is a new phenomenon that we discovered, partly because of some thinking I had done earlier, and it involves also an invention of a new device that can detect that phenomenon, and that phenomenon is related to consciousness, and that's what I'm going to convince you about. So let's go. So I just want to give you, the first part of my talk would just be my motivation to get why I sort of invented the device and started measuring something that may have something to do with consciousness. And the reason is I've been impressed by this notion that consciousness is a serial process, that each conscious moment has limited information holding capacity as it transitions sequentially from one moment to the next. And this notion is as old as David Hume and Alfred North Whitehead, and then William James popularized it in his classic. And then a lot of people have written about it, including Oliver Sacks in his book, River of Consciousness. And that sort of impressed me because of how much has been written about this issue. Therefore, I thought that one could model the underlying brain activity that codes for conscious experiences as a time series of unfolding activity patterns. So the main questions that I have been interested in, as most scientists who are sort of trained from a traditional medical and neurobiological background, is what's the physical nature of consciousness? What's the nature of the neural physical code, if there is such a thing, of qualia? And the most important sort of practical questions that one can address, although they seemed far off in terms of whether we can do it or not, is can we measure the level of consciousness in any person? And can we know whether, and I'm sorry about the typo there, can we know whether an organism is conscious or not? And then finally, does the physical mechanism of consciousness have causal significance for brain function? Because that would be important from an if it is adaptive advantage is what causes us to develop consciousness. And remember, I was thinking in the old traditional neuroscience point of view, right? So what I'll try to convince you, and I think I can probably give you quite good evidence that we can do the 3 and 4 items. We can answer question number 3 and 4 quite satisfactorily from my standpoint. That's what my bottom line is going to be. But potentially also give you some insight, we think, into what's the physical nature of consciousness, and does the physical mechanism of consciousness have causal significance for brain function? Okay, so about 20 years ago, more than 20 years ago, I published the traditional neuroscientific kind of theoretical model, where I thought, in the vein of thinking about consciousness as a serial process, I said conscious experiences are generated and coded by small, non-arbitrary, universal subset of spatially localized neuronal activity patterns. And I was thinking about neuronal electrical activity. And that these patterns, it's a subset of patterns that are mathematically related to each other in some way and are distinct from all patterns that are unconscious. And these are temporal patterns because consciousness is a serial process. These patterns are repeated multiple times over short sub-second periods because of network oscillations which are there in the brain. And the intensity of their associated experience would then be a function of the number of repetitions of those patterns. And the repetitions, as we all know from Hab onwards, can cause consolidation within the brain because of synaptic potentiation. That would be the underlying basis of memory according to traditional neuroscience. And the neat thing about having sequential patterns like that is that limitless variations of these sequential combinations produce the wide range of diverse conscious experiences that we have. So it's a code with limited number of coding elements, but by combination, you can have a diversity of billions of possible conscious experiences. And this has a nice correlate in terms of the genetic code, which codes 20 amino acids, triplet codon, 64 codons, but it can code for virtually unlimited number of proteins. So again, in terms of inspiration, I read Sir Roger's book, Emperor's New Mind, which inspired a lot of the people here who were thinking in terms of quantum physics. But the one other kind of thought that really impressed me was that one by John Archibald Wheeler, who's kind of the pioneer of quantum physics, who didn't get the Nobel Prize because, he was Richard Feynman's mentor and Hugh Everett's mentor, so he's intimately connected with the main idea in quantum mechanics, which is the collapse of the wave function. So he said, no phenomenon is a real phenomenon until it is an observed phenomenon. And I agree with him, although he said it in a different context. He was talking about measurement problem. But when you get to the end of my talk, you'll realize it has an ironic meaning based on what we discovered. So I'm not a physicist, right? Certainly not a quantum physicist. And so I can speculate wildly about physics without being constrained by the rules of physics, quantum physics. So I thought, I took leaps of faith. And I said, what if the coding temporal pattern is actually a pattern of increasing and decreases of wave function collapses? It's not electrical activity, but it's just collapsing patterns. And the antecedent to that would be what Sir Roger talks about in terms of spontaneous collapses occurring. Of course, he has a specific hypothesis. I don't necessarily subscribe to that or try to convince you of that, but definitely what we discovered ultimately has bearing on what he had to say. And the second thing, which gets to the point of the causality issue that I mentioned, the process that causes consciousness actually produces causes something in the brain. So it actually has causal significance in the brain. And that would be, what if this pattern at a given focus or epicenter within the brain can spread in an expanding volume of space in the body and even outside the body by inducing collapses of wave functions in the surrounding region, replicating the original collapse pattern that happened as part of the conscious moment. So that would be a quantum universal temporal pattern hypothesis, because wave functions in the surrounding medium would replicate the collapse pattern. The spatial spread of the effect would fall off with distance, and collapse patterns may be repeated several times, just like what I said initially with the electrical pattern when I was thinking. So these two axes, distance versus time, shows the diminishing collapse probability in the distance as well as time direction. And so you can see that if there is an epicenter within the brain here that spreads out and it extends beyond the brain and the head. So if that's the case, you can if you had a device that would measure that, right, a quantum device, then you should be able to actually test the prediction that there is a spread of this collapsing, this mechanism. And the device would have to be a standard quantum physics device, and which, of course, I knew from, that the double slit apparatus would be such a device where you have waves being converted into particles, and this is well known, it's been well established. I'm not talking of the observer effect itself, but the fact that if light coming out of a laser diode is a particle, then you would see two bands on the screen. But if the light coming out of the laser diode are waves, then you would see an interference pattern. So you can nicely, there's a dichotomy, nicely detect whether it's a wave pattern or a particle pattern. So now if you make a device like this, you have a laser diode that shines light through double slit, and then you have an array of photodiodes that samples the intensity of light falling on them. Then you should see the off-center photodiodes should show you a modulation. If the light emitted collapses into photons, then you should have reduced intensity in the off-center photodiodes, and you should be able to measure that, record that as a time series, right? And that's shown in the two cases there shown. But in addition, you don't need a double slit apparatus to get this effect. You can have a simple laser beam shining light. You have a central beam, and then you have diverging or diffracting beams that are in the surrounding, sort of like a penumbra. And if you have your photodiodes in the penumbra region, the variation in intensity would sort of reflect collapsing wave functions. If the photons, the waves become photons, or particles, that is, they will all go straight down. The ones that are, if the wave, they remain waves, then you should see diffraction and should see greater intensity in the penumbra region. So we made the device. We made both the double slit device and this device, which is basically a laser beam device. And the device basically has a controller, the sensor unit, which has the laser diode and the photodiodes. And then there is an A to D converter. So it's a very simple device. It basically measures the intensity in the off-center photodiodes. And you can record it on an iPad or Android phone, or you can record it on a computer, on a laptop computer. So it's very portable, right? Now, if you do this and you keep the device in the center of the lab, there's nobody in the lab, or they're far away from it, you get what looks like a time series that is flat like that. Of course, there's an initial dip. I can tell you what that is about. But you get, there are 4 channels, 4 diodes, so you get inner bands and outer bands for 4 time series. If you do a principal component analysis of the 4 channels to pick out the variation that is common to all 4 channels, the first principal component gives you that. It's more than 97. The variance is covered by more than 97% by the first component. And you can do a running average. You get a nice, smoother looking curve. I sort of have a convention to display it in an inverted manner. So I subtract the first time point, subtract the first time point from all other time points, so it gets inverted. And so you get a nice curve like that is flat. Now this is when nobody is in the lab, or they're far away from this device. But if you bring the device close to your head on one side, say the left temporal lobe, part of the head, you get a nice little curve that looks like an excitatory postsynaptic potential. In my old life, I was doing intracellular recordings from neurons, and that shows you an EPSP, basically. But this is in a whole person. It's not. So, and it's very reproducible. So you can see that, this thing goes up. It's sort of an alpha function with a normalized trace. Okay. So we found out that this effect is very robust. It is not due to some of the simple things that you might expect. It's not due to body heat, because 37 degrees path does not produce a response comparable to this that you get with your head when the head is close to the device. Ultra-weak photons, biophotons, are not responsible, because no response is seen when the laser is turned off. Respiration, sounds, waves of air pressure are not responsible, because the response is undiminished in vacuum. When the device is in vacuum and you bring the head close to the wall of the vacuum chamber. Radio frequency waves are not responsible and don't interfere with the response, because it's not blocked by Faraday bag if you put the device in a Faraday bag. Magnetic field is also not due to any magnetic field emanating from your brain, because the response is not blocked by a magnetic shield. Electric field, it's not because of an electric field coming out of your brain, because the response is not completely blocked by the metals that we have studied, which should, aluminum and others. So this is just an example to show you, I'm telling you, I have actual data to show. So in vacuum and air, there's no difference. In fact, there's a slight increase in vacuum, in fact. And that shaded portion is actually the duration for which the person was close to the device, about 10 minutes in this case. Most of the time I use 10 minutes. So we did a double-blind experiment. Are we fooling ourselves, or is the person somehow biasing the response? So we basically did a classic double-blind experiment, whereby we had 2 devices. One was active. There were 2 chairs in a room, in a closed room. The one device was active. By the way, I call it a sentiometer, because I believe that it measures sentience or consciousness. So active sentiometer was on one of the chairs, and an inactive one was on the other chair. And the subject randomly, without the knowledge of the researcher, because the researcher was outside the room, sat on one chair or the other, and then we were recording. We could only record from one of the devices that was active. And so whenever the person sat in the chair close to the active device, you saw that response. When it was, he was sitting on the opposite chair, there was no response. There was very little response. Okay, then of course, sentiometric response, which is what I call it, declines with distance from the head. These are really low-hanging fruit type experiments you can do once you get this kind of a response. right? So, and it falls off pretty quickly. By 15 centimeters or so, you have very little response that is barely distinguishable from the baseline. But hold that thought, because I'm going to show you some additional data that might surprise you. We found small differences between where we had the device. On the left side, left temporal side, the response showed the highest peak. On the right temporal side was slightly smaller, but on the forehead, it was much smaller by at least 30–40%. Okay, so then how is it related to consciousness? So the easy experiment for a neuroscientist like me, and I'm in the Department of Psychiatry and Neurology, so would be to do a sleep recording. So have the device 15 centimeters from the head of a person who is sleeping, and you see these nice oscillations. And now those oscillations don't necessarily correspond to the sleep cycle. They are actually shorter in duration to each sleep period cycle. But definitely, every time we record from somebody who is sleeping, we see these rhythmic oscillations. That sort of is suggestive evidence that has to do something with consciousness, because sleep is a phenomenon related to consciousness. Okay, so the other low-hanging fruit is record from all animals, right? And plants, which I'll show you next. But of course, mammals, vertebrates, we had a primate and a rodent. We want to record from a frog and fish and everything, but it is hard to do that in the lab so far. So, right? So we went to invertebrates, and it turns out, with first invertebrate, which is a crayfish, it was in Texas, in Louisiana, you get these crawfish, right? So they're very tasty. So I could get them, and we got that. And turns out that some of the crayfish showed a positive response, just like mammals. But some of the other, out of 5 crayfish, 3 of them showed the positive response. 2 of them showed a negative response. Initially, I dismissed that because I said, what is this, right? So it's the opposite of consciousness, if this is measuring consciousness. I mean, I don't know what that is, right? But then we got blue crab, again went to the fish market, and got blue crab and cherry stone clam. And every single one of them showed a negative response, right? And then we got these tiny worms, California black worms. They're called Lumbriculus variegatus, their scientific name. Oh, sorry. Oh. What is it? I don't know how that happened. Anyway, so if we did that for 15 minutes with the California black worms, we didn't see much of a response. It sort of hinted that there was an inverted response, but so because these are tiny worms, right? So you have to have a whole clump of those worms to actually get a response, and we have to do that for 60 minutes rather than 50 minutes. So, but you get a nice negative inverted response so pretty much all invertebrates, I think, show the opposite of what vertebrates show. So it has some kind of inverted consciousness or something, which of course nobody would be able to tell. So anyway, we did plants. Potted plants are easy to get, did the thing. So initially we did 15 minutes exposure, and there was some tendency for it to be an inverted response down, but in the top part, you see kind of a flat response. So I thought initially that there was no response, but then I did for 60 minutes, and you definitely see an inverted response. So plants are more like invertebrates in this way they, again, we don't know if that is consciousness or what kind of consciousness that is, or is an opposite of consciousness of some kind. So anyway, so then, obviously, the next step would be what Stuart would want to do, which is do anesthesia, general anesthesia in these mice. And so now we have studied 3 anesthetics. We are going to study 3 more. I don't have that data yet. But we started out first with actually isoflurane. and then ketamine xylazine. Right, that's the mouse anesthesia. And these mice, I mean, it's a standard procedure, approved by our Animal Protocol Committee. And then what you see is when you anesthetize the mouse, first obvious thing you see, and of course you have to do this for an hour. If you do it for 15 minutes, you see a distorted response, which is what I presented at Taormina, but that wasn't the full story. So what you see is the amplitude of that response is much reduced, the peak amplitude. But one other thing that you notice is actually the rise time. So the response occurs much faster in anesthesia than without anesthesia. So the rise time is shorter. So that's plotted there statistically. I mean, you don't even have to do statistics on these things. It's so black and white, the whole thing. Okay, so but the neat thing that I observed, which was surprising, was that the amplitude was not very determinant of recovery from anesthesia, or amplitude does not determine the level of consciousness. What determines it is the rise time. The rise time is shorter, which if you can see those graphs there, the recovery occurs of the rise time occurs better than of the amplitude. In fact, with ketamine xylazine, even though the mouse was moving around, the amplitude was still low. What had recovered was the rise time. And so what is this? What's going on? So what I thought immediately was, the one interpretation of this would be that, 5 minutes? Oops, sorry. One interpretation of this would be that in the awake state, because you're producing structured patterns, it is more desynchronized. Whereas in the anesthetized state, you get a synchronized synchronization of the response. And this is kind of a numerical simulation. So I look at it, I mean, you can look at it from a microtubule standpoint, but there are sites at which collapses occur if you use the collapse interpretation. And those sites are collapsing one at a time. Anesthesia knocks out some of the site, but in the sites that are still active, they are more synchronized. And that's why your rise time reduces and the amplitude reduces as well. So I have to apologize for the next couple of slides, and that's what Thomas told me to do, because this is insensitive to some people. So I apologize for that. So I am killing mice. So, okay, using standard humane methods that are approved by our IAQ committee. So it turns out, though, the response persists after death, okay, in these mice. And you see the same thing. They get synchronized. They're in the awake, alive state, they're desynchronized. In the awake state, in the dead state, state of death, they're synchronized, okay? So I have to also apologize for the next thing, which is, of course, these are the dead mouse. I want to separate the brain from the body, because, according to my hypothesis, there is a spread that originates, there is a source that originates in the brain, and it spreads to the rest of the body. So you can detect this effect out. That's why I call it Peri-Somatic. You can detect it in the rest of the body as well. But the brain is able to do it actively, modulate it actively. I wanted to see if that is possible. So we cut off the head of the dead mouse and did that experiment again. And it turns out that the body just is a passive vessel that basically takes whatever brain made and just passively decays down. But the brain, the head in this case, after that, modulates the response. So initially you get a biphasic response, like up and down, and then about 3 hours after the death, you get only the negative response, the inverted response, just like an invertebrate. So when we die, I guess we become invertebrates for a while at least. I don't know how long this lasts. I suspect, because we can detect it at 5-6 hours in a mouse after death, so I suspect in humans it might last for days. I'm not sure. Yeah, so one other here example where there is modulation by the brain. Here you see sleep-like patterns in the positive response, done only by the head, not by the body. Okay, so this is the last thing I wanted to show. So what determines the baseline, right? So it turns out, so we did an 18-hour experiment. At 3:30 in the afternoon, we turned on the machine, the device, and then let it go until 9 o'clock in the morning, or 9:30. And it turned out we saw this kind of a response. So when the researchers were leaving the lab, the response came down and settled on some low level. And then when the researchers returned to the lab, and they were far away, then they were not close by, so it went up. So what is it? So I think there are two things going on. This is the hypothesis. The consciousness is generated by a molecular lattice, probably microtubules, but it could be mitochondrial network as well. Who knows, right? And those are the ones that code for qualia. And then neuronal electromagnetic activity orchestrates, orders, or assembles these patterns into larger functionally effective patterns. Left on their own devices without electromagnetic control from the brain, the waveform collapses tend to synchronize, which is what I showed you. And the collapse patterns themselves exert collapsing influence over the quantum wave function occurring in space and within neuronal networks in the brain. So there's a bidirectional control, which is what I show you. So here's bidirectional control from the electromagnetic activity in the neuronal networks, and temporal wave function collapses in the molecular network. So general anesthesia knocks out some of the molecular sites, and it also probably shuts off the bidirectional control. In clinical, the brain electromagnetic activity is shut down, but the molecular activity can still go on because it's a bioenergetically independent process. It's a quantum process, probably. So, and then I predict that in psychedelic state, we would partially knock out the bidirectional. This is what we are doing next, trying to mine qualia-related patterns from the brain with a new device, which is based on this device. And these are my research colleagues who did all the experiments and made the device. Thank you very much for your attention. Sorry for the...
Thank you so much. Is this on? No.
Oh.
Okay, yes. How about one quick question, and then we'll go, and we'll have questions for both of them after Dean Radin talks about a similar topic.
Yeah.
Did you actually interrogate any inanimate objects like a block of steel, a rock? How about a power motor or a blender or microwave oven?
If you turn that on, so heat. If you turn that blender on, it generates heat. So you see a small deviation. It's never as much as you see with, any kind of heating device produces I think it's because it changes the sensitivity of the device itself.
There's no electromagnetic
No. Yeah.
We're going to hold the rest of the questions until the end, and we'll do some both with both
Yeah.
of them. Let
Okay.
Dean